Hi everyone. During today's live session, we're going to be talking a little bit about meiosis and how it relates to sexual reproduction. But more than anything, to be able to understand meiosis, I need to take you guys back to mitosis. Okay, so we're going to be really focusing on mitosis so that later on during next week and so on, you'll be able to understand meiosis and how it relates to sexual reproduction in human beings. So let's start off with mitosis, okay? So mitosis is cell division. We use it as cell division, and there's organisms that use it as asexual reproduction. That means that they pretty much clone their own cells and they do not need a second partner to do this. So they're simply reproducing by cloning their own cells. So what is mitosis? Mitosis produces cells with the same exact information. So they produce identical daughter cells or clones, okay? They're exact copies. There's absolutely nothing different between the original parent mitosis cell and the daughter cell. They're exact copies or clones. And it also produces a cell with the same amount of DNA, same number of chromosomes, same genetic information. So again, if you can think about mitosis as cloning, that is perfectly fine. In some organisms, this form of cloning is how the organism reproduces. So there's certain single-cell eukaryotic cells and very, very simple multicellular eukaryotes. So we are multicellular eukaryotes, but some very simple organisms, multicellular organisms, can reproduce asexually, or they can produce clones of themselves to create new organisms. This is a little organism called a hydra. This is yeast, okay? And this asexual reproduction in these organisms is called binary fission. So it's mitosis, but since the goal is to reproduce, they gave it a new term called binary fission. We cannot reproduce through binary fission because we're not producing clones of ourselves when we're reproducing. We're producing new genetically different organisms. So what are we doing? If we want to reproduce, we need to join two different cells, two different sex cells in human beings. We do that by joining the eggs of females with the sperm of males. And when we are making egg and sperm, they're not genetically identical cells. So we're not doing this by mitosis. We actually do this by meiosis. We're going to talk about meiosis later on. So let's just look at it super briefly. A normal human cell is going to have 46 chromosomes. Why? Because 23 of those chromosomes is given by mom, 23 of those chromosomes is given by dad. So every single cell in your body contains a total of 46 because you're made up of mom and dad. Okay? But to originally make you need to have one cell that has 23 chromosomes, and then you need to have another cell that has 23 chromosomes to eventually make you, which you are a total of 46. So to produce these 23 chromosome cells, in this case, if it is the female, she is producing an egg, and eggs are 23 chromosomes. And if this is the male, he is producing a sperm, and a sperm is 23 chromosomes. What you need to do is you need to half or cut in half the original cell, because the original cell has 46. So you cut it in half, making 23, and eventually these two cells that are 23 both will merge in something that's called fertilization and will eventually make a new human being. So we're going to check that out. This is going to be a little bit confusing for a while, okay? So do not get impatient. We'll continue checking this out later on. Let's first of all review mitosis. That's what our goal for today is. So when you're talking about mitosis, you're talking about the division of a cell to produce two new cells, okay? And usually, what is the purpose of mitosis, specifically in human beings, for example? We're constantly doing mitosis to grow, and we're constantly doing mitosis to repair our cells. Sometimes if you get a cut, for example, in your skin, you are damaging a lot of those skin cells. So how can you repair them? Well, some of those skin cells will go through mitosis, producing new cells and repairing those original damaged cells. So there's 4 stages of mitosis. Maybe this starts ringing a bell. Okay. The stages of mitosis are prophase, metaphase, anaphase, and telophase. Okay. Prophase, metaphase, anaphase, and telophase. Phase stands for stage. Okay. And we're going to talk about those stages a little bit more right now. Okay. If you want to see it in a graphic, what you can see here is that the cell will start going through prophase, will then continue metaphase, then will continue anaphase, and finally telophase. And there's a little tiny stage at the very end, which is cytokinesis, which we're going to talk about in a second. So why does the cell go through all of these stages? Basically, it goes through these stages to produce 2 new cells that are identical to the original cell. What does that mean? If every single cell in your body contains 46 chromosomes, that means that your cells are diploid. Why diploid? Because they contain 1 set of chromosomes from mom and 1 set of chromosomes from dad. Each set is made out of 23 chromosomes. So in total, you contain 46 chromosomes. You contain 2 sets. This N here stands for set. Okay, 2 sets of chromosomes, 1 from mom, 1 from dad. So you want to produce cells that are also diploid. You want to produce new copies, clones of this cell. So you want to produce, again, diploid cells. Okay, so you're going to be doing that through prophase, metaphase, anaphase, telophase, and finally you're going to break up those 2 new daughter cells by a process of cytokinesis, where you are literally breaking them up and creating 2 separate cells. So let's go into this idea of mitosis. So when your cells are in process of, when your cells are living, okay, any living cell is going through something that's called the cell cycle. So it's just a cycle, okay? And usually when the cell isn't actually doing anything, it's not pretending that it wants to divide. It's not really doing anything. It's just completing its function. Let's say that this is a liver cell. The liver cell is just completing its function. It doesn't want to divide. That's when this cell is in a stage called interphase, which means it's just in between stages. It's really not doing anything. It's just completing its function, and it doesn't have the purpose to divide yet. It doesn't have the necessity, okay? So the vast majorities of cells are just in this interface, not really thinking about dividing. And then at times, something triggers this cell to think about dividing, to think about going into mitosis. So when the cell decides that it needs to go into mitosis, it will begin with prophase, okay? Once it completes prophase, it will go to the next stage, metaphase. Then it will go into anaphase, then it will go into telophase, finally cytokinesis, and now you have 2 new cells. So let's start off with just this idea of interphase, this idea of the cell isn't actually doing much. It is just completing its function, and then when it suddenly decides to divide, it will start just preparing for division. Okay, and when it starts preparing for division, then each one of the chromosomes are going to be doubled. Because remember that you want to produce 2 new cells with the same amount of genetic information. So if you want to produce these 2 new cells with the same amount of genetic information, then you need the chromosomes to be copied and doubled. So from the 46, you're going to double that into 92. Okay, now in prophase, now you've got those 92 chromosomes in prophase, and what's going to happen is that all of those chromosomes were originally in a little structure called the nucleus. The nucleus is just a place that holds all of your DNA, and it is closed up by this little nuclear envelope. So this nuclear envelope and this nucleate is going to start to disappear, okay? And there's going to be little tiny structures, organelles, that are called centrioles, and they're going to actually be the organelles that are going to pull apart some of these chromosomes and allow you to divide it into two different cells equally. And these centrioles are going to start moving to opposite poles of the cell, because at the end of the day, the centrioles will be moving the DNA to opposite poles of the cells. So centrioles start moving to opposite poles. So these are the centrioles that I was showing you guys. In prophase, they're really not that apparent, but in the next stage, they really are. So there are these little structures, organelles, that are going to eventually pull the genetic information to opposite sides of the cells so that you can get two cells with the equal amount of information. In metaphase, a look at the M, okay, the chromosomes are going to line up right in the middle, okay? So now you've got 96 chromosomes lined up in the middle, and 46 are going to want to go to one side, and the other 46 are going to go into the other side. Remember that these 96 are just 46 copied, okay? So 46 doubled. So they're two completely identical sets of 46. So one identical set is going to go to one side, and the other identical set is going to go into the other side to create two completely identical new cells. And in metaphase, these chromosomes line up in the middle, right in the middle. That's why it's called metaphase. In anaphase, one set of 46 is going to be divided from the other set of 46. So they're going to be split up. And when they become split up, the centrioles are going to start pulling them apart towards opposite ends of the cell, opposite poles of the cell. So the chromosomes are separated and they're going to be moving towards opposite sides of the cell. That's anaphase, when they start this movement towards opposite poles of the cell. And then telophase is when each side of the cell now contains the original 46 chromosomes, 23 from mom, 23 from dad. So you can start reforming the area that holds this DNA, which is called the nucleus or nuclei if it's in plural form. So you're going to be reforming two new nuclei, okay? And the only thing left to do is actually split both cells up. And the splitting of these cells up is called cytokinesis. So you literally split the cells up to form two new completely independent cells that are identical one to the other. So the cell membrane moves inward. So this right on the outside is called the cell membrane. And what it does, it actually starts moving inwards from this part. If you can see it here, it starts moving inwards until they join together on both sides, and there's a complete separation of these two new cells. Each new cell contains its own nucleus and contains 46 chromosomes, 23 from mom, 23 from dad, and they're completely identical to the parent cell. So to remember this, just think about interphase, prophase, metaphase, anaphase, telophase, cytokinesis. This is just something that some teachers use to help you memorize. In practice, my aunt teaches chores. Okay, if this doesn't work for you, that's fine, but it does help you memorize the order. Okay, remember, interphase is not part of mitosis, it's just a cell preparing to divide, while as all of these stages are part of mitosis. And here you can just see some mitosis in real life cells. Interphase, just preparing for division. Prophase, the chromosomes have been duplicated. Now you have 92 in total. Metaphase, the chromosomes have lined up in the middle. Anaphase, the chromosomes have been divided. 46 are going to go to one side. The other 46 are going to go into the other side. Telophase, there's a nuclear membrane that can be reformed to contain each of the new cells' genetic information, and the cellular membrane starts to move inwards. Finally, after telophase, you create two new cells, and these two new cells can go back into not really thinking about dividing, just being an interphase. So hopefully that makes sense. And yeah,